Good evening. My name is Dr. Arlene McDougall. I'm the Director of Research and Innovation for Mental Health Care at St. Joseph's Healthcare London, as well as the Parkwood Institute Research Mental Health Theme Lead, part of the Lawson Health Research Institute. I'm also a psychiatrist and a Lawson researcher specializing in recovery-oriented interventions, including mindfulness-based interventions for people living with serious mental illness, as well as other recovery-oriented interventions, both here locally as well as globally in Kenya, for example. More recently, I've gotten involved in the use of social innovation approaches to tackle the many challenges we face in our mental health care system. I am absolutely thrilled to be here with you today and to host and facilitate this wonderful evening. On behalf of the Lawson Health Research Institute, I welcome you to this Café Scientifique entitled When Connections Break Down, Slowing the Decline to Neurodegenerative Diseases. So Lawson is one of the top 10 hospital-based research institutes in Canada. We conduct leading-edge research that advances scientific knowledge and healthcare for our patients, now and in the future, here and around the world. We do this as the Research Institute of the London Health Sciences Centre, LHSC, and St. Joseph's Healthcare London. Both academic health organizations have care, teaching, and research mandates to fulfill. And it's Lawson that oversees the research mandate of each hospital. And our scientists work within hospital walls as well as sites across the city. They are working in dedicated research labs and alongside care providers in clinical areas and partnering with our study participants. This is what is called a bench-to-bedside approach. Our researchers have a desire and a deep passion to continually improve the outcomes for our patients and their families. So our research questions are formulated at the point of care. We ask, are there frustrations? Are there issues that we can address or we can do better? Can we improve a treatment or fill in an existing gap where a treatment doesn't exist? And then once sufficient testing has been undertaken, how do we actually implement our new solutions, our better solutions, to have the best possible outcomes for patients in the most efficient way possible for our system? So tonight, we will be focusing on neurodegenerative diseases. I'm sad to say that in Ontario alone, there's an estimated 285,000 people currently living with some form of neurodegenerative disease. This includes Alzheimer's disease, Parkinson's disease, mild cognitive impairment, other forms of dementia, multiple sclerosis, and many other types. The experiences and symptoms for each person vary, but they do share a progressive decline, both in cognitive and motor function. It can affect their ability to work, to be with their families, to socialize, and to live independently. As our population ages, there is an urgent need to understand markers that will predict this decline and identify targets for therapy that might stop or at least improve long-term function and outcomes. This is where hospital-based research can really make a difference. So in just a moment, you'll be hearing highlights of the groundbreaking research on neurodegenerative diseases happening in our hospitals right here in London. So now I would like to introduce our first speaker, the Lawson researcher, Dr. Jenny Wells. Dr. Wells is the chair of the Division of Geriatric Medicine, as well as an associate professor in the Department of Medicine at Western University's Schulich School of Medicine and Dentistry. Her clinical and research interests are Alzheimer's disease and geriatric rehabilitation. She has a particular interest in mild cognitive impairment and the potential for non-drug interventions, such as exercise, nutrition, and others to slow progression of dementia. She is a principal site investigator in randomized controlled trials of new drugs to treat dementia, mild cognitive impairment, and subjective cognitive impairment. So please join me in welcoming Dr. Wells to the podium. Thank you.
So thank you, Arlene, for that introduction, and not just my personal introduction, but the introduction to Lawson and really how important research is in the hospitals and research that's meaningful to improve people's lives. So I'm going to give you a pretty fast smattering overview. I timed this earlier. It was 14 minutes long. I was told to make it 10. So I'm going to go pretty fast, and you can catch all the for questions at the end. Okay, so we've retitled the research group of Cognitive Clinical Research Group. Got rid of the word geriatrics because we've invited our colleagues in cognitive behavioral neurology, who their name will be up there, and one of whom is Dr. Liz Finger, who's here tonight, to join. And it's part of the Parkwood Institute research branch of Lawson. So that's what I'm going to talk about, a smattering of all the studies that are going on. I'm going to focus in on a couple key points, though, that I'll go slower with that. So it's Michael Borey and I and Dr. Nevo Reagan and Dr. Manuel Montero Dasso are geriatricians. Dr. Finger, Dr. Pasternak are the cognitive neurologists. Dr. Fogarty is a PhD clinical psychologist who assesses specialized cognitive tests. And we have help with biological markers, particularly with neuroimaging and MRI studies with Dr. Rob Bartha and Dr. Frank Prato. And you saw a picture in Dr. McDougall's slides of Dr. Amer Burhan. So he collaborates, and we're so happy to have the neurologists, the geriatricians, and the psychiatrists involved in research at Parkwood at one site, because it's very synergistic, and dementia really needs all three of those types of disciplines to assess all the types of symptoms. And Dr. Manuel Montero Dasso is an MD PhD, and he specializes in gait. And he's in the pamphlets on your book. There's a write-up about his work and looking at gait as a marker of dementia and an early gait changes. Like you've heard of the expression, you can't walk and talk? That's true, and he has research to prove it. So we work on dementia, pre-dementia syndromes, and Dr. McDougall introduced you to the little abbreviations of SCI is subjective cognitive impairment. That means you test normally, there's no impairment in everyday life function, but you think, I'm working harder to get this stuff done. Mild cognitive impairment is you're not only working harder, but your tests are not expected. They're not expected for your age or education level. But your function in everyday life is still normal. Dementia means you have cognitive impairment on test compared to your education and life, and that your function in everyday life begins to become impaired. And there's Parkinson's dementia. I left off, maybe unconsciously on purpose, because we have a Parkinson's expert to speak about Parkinson's disease. But we are doing research in Parkinson's dementia with a very well-tolerated cough syrup called Ambroxol that Dr. Jog and I, led by Dr. Stephen Pasternak, and Dr. Finger and I are all collaborating on. And we do research in frontal temporal dementia. So someone before this talk asked me to explain the word dementia. Dementia is the generic term for neurodegenerative diseases that cause trouble in thinking. Alzheimer's disease is one type, the most common type. 80% of all dementias is due to a combination of Alzheimer's disease and strokes, or one or the other. The other 20% are these more rare syndromes. So we look at our group at Parkwood. We're looking at pharmaceutical company-sponsored research for new drugs, which I'll highlight a couple examples of why we're excited about that. We're looking at imaging and biomarkers because, face it, we don't do brain biopsies. That's a pretty invasive technique to find out if you have a neurodegenerative disease. You know, unlike having a cancer that you can easily stick a needle in and get a biopsy and prove what you have, dementia and looking at the brain is more challenging. So we're looking at specialized biomarkers, whether it's cerebrospinal fluid, to look at the proteins that are produced by the brain that are associated with the diseases, or we have, I think, the only 7T, that's 7 Tesla, is a very powerful magnet, MRI in the country, here in London, and other PET imaging, which the PET scan is a proton emission tomography scan that looks at the metabolism of the brain, and we have special markers that look for the specific abnormal proteins that are associated with dementias. And I'll highlight some of those. Those fancy color pictures in one of my first slides is an example of PET scan. And there's other studies we do with what we call observational outcomes, and that's looking for the progression of dementia and the different types. And it helps with hypothesis generation. And we are networking with the Canadian Consortium on Neurodegenerative in Aging. That's a national study with experts in the country to look and develop questions about dementia and how it progresses, as well as the Alzheimer's disease neuroimaging trial, which is an international trial looking at MRI scans as a biological marker for progression of dementia. So this slide is busy. You can't see it from the back, but that's okay, because it just highlights that we have the whole span from dementia to pre-dementia syndromes. So one of the things that I've highlighted when I teach my students and residents is that Alzheimer's disease, for example, doesn't start the day you become forgetful. The disease starts in the brain with abnormal proteins developing and laying things down 15 years before you have your first symptom. So that some of the strategies are to look at people who are at risk with the late onset Alzheimer's gene. That's called the ApoE4 allele. It doesn't mean you're going to get dementia, but it increases your risk. And for example, the drug trials are focusing on monoclonal antibodies against the amyloid protein, which is one of the proteins involved in the development of Alzheimer's disease. So if we can remove amyloid from the brain earlier, does it change the disease progression? And so there's other things. We're looking at exercise as a modality, and Dr. Borey has initiated, this is not a drug trial. No pharmaceutical company is going to make a lot of money over jazzercise as an intervention to prevent dementia. But we're looking at biological markers with MRI to see if the brain changes in response to exercise. And it does. Even walking can change your brain. Meditation can change your brain structure. So then we have a whole bunch of other trials looking at the other protein called tau proteins that are abnormally deposited in Alzheimer's disease to see if that will change the course of the disease. Oh, the A4. That's a 4-year-long trial for people who have subjective cognitive impairment. They have a first-degree relative with Alzheimer's disease, and they have a brain that's assessed by a special PET scan to look for the amyloid. So they have a brain full of amyloid. Their memory testing is normal or very near normal. Their function is normal, but it's going to see if it slows or prevents the development of dementia in the 4-year timeline. So I've talked about it. Dementia is a disease of the brain that affects memory, thinking, behavior. The overall causes are still being explored, and there's many. That's part of the reason why the treatment for Alzheimer's disease is so difficult, because there's many different types of causes. So the tau proteins might be one theory, and the amyloid protein probably together. Oxidative stress, and that's a hard thing to measure, but the Mediterranean diet will reduce the oxidative stress and create a healthier system in your body, for example. And other immunological factors. For example, head trauma, poor diet, low education, low level of stimulation is also associated with the development of Alzheimer's disease, as well as certain genetic dispositions, or I should say predispositions. So we don't have a cure, but we're going to find something that's disease modifying. So this is my chance to influence your life for the better. The Mediterranean diet prevents heart attacks, strokes, and Alzheimer's disease. Take one thing home, eat that way. And the other would be to exercise. And there's in Dr. Manuel Montero's Adaso, there's something about walk the walk. So I try to walk the walk and talk the talk. I moved my gym appointment early so I could come here tonight. I did. Okay. So this is for the auditory learners, so this is a PET scan, and they looked at people who have that late onset Alzheimer's gene. And I would rather show you this slide because it's visual. So the people who have the gene, the ApoE4 gene, who don't have dementia, they don't have any complaints, but they have the gene, and they're non-exercisers, have a whole lot more amyloid in their brain than the people with the gene, same risk gene, who exercise. On the left in green and yellow are the people who have the more healthy type, the non-ApoE4 genes. So if you exercise with this risk factor, and that's why we don't recommend genetic testing for the ApoE4 gene, because everybody should be exercising and eat a Mediterranean diet. So I wanted to show you that exercise is an important treatment for health and brain health. And then this represents the jazzercise intervention. The pilot is underway with just 9 folks, and that will hopefully lead to a larger trial where we're looking at we're measuring commitment. They're supposed to go to a jazzercise class 2 or 3 times a week. We're measuring their compliance with the class by their Fitbit, which is right here. I count my steps. At least 10,000 every day. Sometimes I'm at my bedside doing this to make sure I get 10,000 in. And then we're looking at their level of fitness. We're going to eventually, in the larger trial, get the gait studies with our colleague, Dr. Montero Adasso, and look at their balance, because balance is really a function of brain function also, and look at their MRI scans. That should be disease modifying. We're going to do cognitive tests as well. This is an example of how many research trials over the last basically 15 years have failed to be successful. So down here is 0.5% have resulted in drugs on the market for us to use. The ones on the market are not disease modifying. They just modify your symptoms a little bit for a short period of time. So hepatitis C is 2%. The Staph aureus, 4.6% new antibiotics. 4.1% is the industry average. So Alzheimer's disease has been a tough disease to research and be successful with. So I already commented on this. The drugs that are currently on the market aren't disease-modifying. The new drugs that I'm going to give you a snippet of, we think they are disease-modifying, and I'll show you why. And so it's aiming at changing the actual disease process in the brain by reducing those amyloid proteins and the tau proteins that accumulate. And then why did they fail is one of the questions. Is in the trials that we did 10 years ago or so, we didn't screen the patients with the amyloid PET scans. So there is probably some uncertainty whether this patient had Alzheimer's disease. When we did trials with people with mild cognitive impairment where a person is a little bit forgetful, but they don't have dementia, can last 8 years. So if we got someone in the beginning stage or the late stage, and the trial was only a year long, they're not going to progress. So therefore, the trial might be negative when it could have been a decent drug. So it's challenging. Okay, I'm going to skip this because this is just a cartoon picture of the amyloid protein and evolving over time. It's a 42 amino acid long, the A beta that's cleaved from the large amyloid molecule. The A beta 42 is the one that's more likely to clump and form polymers and oligomers and then form the plaques that are associated with the disease. This is a picture of the tau protein. So you know, if you've had anyone take high school biology? Most people. Okay, good. You have an image of a cartoon of a neuron with a long tail that sends the messages down to the other neurons to communicate. It's this tail that has the communicating train tracks. That it forms the tangles. When this brain cell dies, the axons, the train tracks crumble, and they're left with these train tracks. So looking at, there's enzymes that can hopefully block, and when this tau is phosphorylated, then the train track crumbles. So we're trying to block the breakdown of those axons. I was talking about the PET scans and measuring the amount of amyloid in the brain, and that some of the new drugs called monoclonal antibodies, they've been various types of failures of them over the last 10 years, but we're having some that are becoming winners. In this particular one, this is a person who's been treated with a monoclonal antibody. And you can see at the beginning of the trial, there's more red, and after 78 weeks of treatment, there's less red in this brain. So the red is the amyloid protein. This one, it's more obvious. red amyloid buildup, less amyloid after 78 weeks of treatment. In the bottom two are two people who are on the placebo for this trial. They have red on the left and even more red after 78 weeks. So there's more amyloid buildup. So what's important to me is not just, theoretically, does the amyloid less? What does it mean in real life? So the measure we use that reflects clinical responsiveness is called the CDR, some of the boxes. It looks at, it's caregiver informant based as well as cognitive test, clinical dementia rating scale. So we look at that in terms of judgment, insight, planning, functional task, and the placebo group deteriorates, gets worse. The two groups of the highest dose are overlap here, this red and this sort of blue, are successful. The CDR is lower. Lower is better. So this is clinically different. So it has a real effect on a person's behavior and thinking. So that's some of the last slide was about a pharmaceutical company-sponsored trial that we're participating in. And so now, a little bit of a sales pitch. Why would you or your relatives want to participate in a clinical trial? In some cases, they actually might help you. And I would dare say that just participating with a group who cares about you, who shows an interest in you, who monitors your health, and provides you with education, can be supportive emotionally. So there's some benefit that way. In the trials, we're very careful about monitoring for other health problems you might have. So there's an education effect. There's close medical monitoring. There is certainly a placebo effect that is proven to be beneficial in trials. And when the trial is looking like successful and coming on the market, so for example, donepezil, galantamine, rivastigmine, and memantine. Over the years, starting in 1998, Dr. Bourret and myself were in those phase 3 clinical trials that led to those drugs being put on the market. And my patients had access to them before they were on the market for regular prescription because of the open-labeled extensions. So that's a benefit. The other benefit is altruism. You're advancing science, and it might help someone else. So to conclude, we have a list of all the support staff that help communicate with my patients and with the research subjects and run the trials and tell me what to do. And I will thank them and thank you for your attention.
Thank you so much, Jenny. And I think we all now have permission to go and have some red wine and some more goodies. I'm thinking, why don't we just package, red wine and some olives in a pill form and go with that. So we'll save our questions. So jot them down, your thoughts, your comments for Dr. Jenny after the other speakers. So our second speaker is a Lawson researcher, Dr. Elizabeth Finger. Dr. Finger received her medical degree from Cornell University. She completed an internship in internal medicine at Massachusetts General Hospital, followed by residency in neurology at the General Hospital and Brigham Young and Women's Hospital in Boston. Her research focuses on understanding the cognitive, so the thinking, the neural, the nerves, and the genetic substrates of abnormal decision-making, emotion, and social behavior. She uses a variety of different diagnostic techniques and modalities, and her research program investigates the cognitive and neural systems affected in patients with frontotemporal dementia, related disorders, as well as their at-risk family members. So please join me in welcoming Dr. Finger to the podium.
Thank you, Arlene. And I realize with the description of our research, it's appropriate to say much of our focus for the past 10 years while I've been in London has been on social cognition and looking at remedies for that in some of the dementias. Tonight I'm going to talk about a topic that's a little newer for our group, but is quite compelling, and I think you'll see why we've become increasingly interested in this. Although the caveat then is I have no answers, but I have a lot of questions to pose for you. So neuroinflammation in dementia is an old idea, as you'll hear. It's been around a few decades, but there's been a resurgence in interest in how inflammation in the brain and the neurodegenerative diseases that you just heard about from Dr. Wells may be related. I'm going to back up for one moment to the big question of what causes dementia. And thank you, Jenny. You set this up in a lovely way for me. So this will be a review for you now if you didn't know already. In Alzheimer's disease, as Dr. Wells mentioned, the main two molecules that we see building up in the brain are called amyloid and tau. They build up outside of the synapses, in the neurons, and it's thought that these two molecules are toxic to the neurons, causing them to die, and eventually when enough neurons die, we have cognitive symptoms or deficits. That was known for about 100 years, but more recently, some of the main genetic mutations that cause familial Alzheimer's disease were identified, and these clustered as they were all genes that involved the amyloid molecule, either coding for the amyloid protein itself or some key enzymes that modify that protein. And so the dominant hypothesis as to the cause of Alzheimer's disease focused on the abnormal accumulation of amyloid in the brain, followed by tau. And thus much of our therapeutics, as you've heard to date, have singularly focused on trying to reduce amyloid deposition. In frontotemporal dementia, another neurodegenerative disease, we see tau as well building up again in ways that it shouldn't, although just in a slightly different shape or conformation, or sometimes a protein called TDP43. And in a sort of parallel model, when one of the first genetic discoveries was made of a genetic cause of frontotemporal dementia, that gene happened to be MAPT. That's the gene that codes for the protein tau. So this fit nicely with the models of Alzheimer's disease, and it was thought then that the accumulation of tau must be driving the toxicity to neurons of frontotemporal dementia. And much of our therapies, although many decades behind the science of Alzheimer's disease, are currently focusing on trying to remove or reduce tau deposition. In Lewy body disease and Parkinson's disease dementia, which I'll touch on, but we'll leave to Dr. Jog for the third part of the evening, we see a different molecule building up. This one is called alpha-synuclein. It clumps up in nice round balls in the cells, but leading models suggest that that alpha-synuclein may be toxic to those cells. While mutations in alpha-synuclein are not a common cause of Lewy body disease or Parkinson's disease, much of the therapeutic focus has been on trying to reduce their accumulation. With that, and when we finally have some promising amyloid therapies, though, as you heard from Dr. Wells, there have been many unsuccessful trials. So that and some other happenings in the field have led to some more creative thinking about is amyloid or is tau or alpha-synuclein the only part of the problem. This slide says immune cells are also present at sites of dementia inclusions. So we've also known for many, many years that in addition to amyloid or tau depositions in the brain in dementias, we also see increased numbers of the immune cells of the brain. These cells are called microglia, and they're present in elevated numbers. So this in black is compared to controls, the number of these microglia or immune cells. We see increased numbers in brains of patients with frontotemporal dementia, here in red, or elevated as well in Alzheimer's disease, and the same is true for Lewy body disease and Parkinson's. Now, people knew this for a while, and they didn't think it was necessarily that exciting, because one thought was, well, we know the cells are dying, other things are causing them to die, and part of the normal role of these microglia is just to clean up the debris and clear it out when it's something that the brain isn't needing anymore. Others, though, said, well, maybe they're not just the garbage collectors. Perhaps they're actually a part of the pathology. So with that hypothesis, then, the epidemiologists had a specific idea. They said if these immune cells or neuroinflammation is actually part of the problem, then perhaps in people who have taken anti-inflammatory medications for a while medications that would dampen down those microglial cells and dampen down their activity, perhaps we would see that they were less likely to get Alzheimer's disease. So many focused on people with rheumatoid arthritis. Rheumatoid arthritis is an autoimmune arthritis. The immune system in the body, not in the brain necessarily, but in the body, is revved up. And so patients with rheumatoid arthritis, especially back in the 80s and early 90s, were mainly treated with anti-inflammatory medications like ibuprofen, aspirin, steroids, or other non-steroidal anti-inflammatories. When the epidemiologists then looked at this, and this is a meta-analysis looking at 17 of these studies, they thought, yes, indeed, it looks like patients with rheumatoid arthritis may have a reduced risk of developing Alzheimer's disease, and so we will infer that it's these anti-inflammatory medications that may be conferring some protection. However, that's an association. It's certainly not proving cause. There could be other confounds that are giving you that pattern. So the next step was to try randomized controlled trials, the gold standard of proving that a treatment works. So then came many clinical trials. These are published in very high-impact, respected journals, as you can see. And unfortunately, essentially, they were all negative. So they looked at people who had MCI and were at risk of converting to Alzheimer's disease, but different anti-inflammatory medications did not reduce that risk of conversion. They looked at individuals who were older and had a family member with Alzheimer's disease, so their risk was high of progressing. And across all of these studies, none showed any benefit of the anti-inflammatory medications in reducing the risk of dementia or in slowing the rate of cognitive decline. So, needless to say, that was a bit of a downer for the field, and people refocused again then on the amyloid and tau hypotheses. So that was put aside for a while until some new technology, specifically in the realm of genetics, allowed for kind of the first time widespread sequencing of people's entire genome, so looking at every single gene that they possessed in large numbers. What happened then, sorry, just to see if this will help. Yep, so new genes related to the immune system were identified that could modulate the risk of Alzheimer's disease as well as some of these other neurodegenerative dementias. ApoE and ApoE4, as you heard from Dr. Wells, is the most common genetic variant that increases the risk of developing Alzheimer's disease. It doesn't appear to cause it itself, as you saw from Dr. Wells, but interestingly, the ApoE gene is the protein that it codes for is mainly expressed on the reactive microglia, those immune cells in the brain, as well as the astrocytes, which help to support the other cells and neurons in the brain. Another big discovery was that mutations or variations in another gene called TREM2 increase the risk for frontotemporal dementia, Alzheimer's disease, and I believe Parkinson's. That gene is only expressed in microglial cells in the brain, so something about it that's changing the way its protein functions seems to be conferring either an advantage or disadvantage to develop these conditions. Of great interest to us because of our focus on frontotemporal dementia, the third one there, the CSF1R, was another mutation discovered not long ago. It can cause a form of progressive neurodegenerative dementia, which clinically can look like frontotemporal dementia or Alzheimer's disease. That gene codes for the receptor on the microglia that's critical for the survival, the growth, and the function of those cells. Again, really strong signal to say the immune system is popping up all over in terms of modifying the risk of these conditions. And finally, progranulin. Back in 2006, this was discovered as one of the most common genetic causes of frontotemporal dementia worldwide. But as that discovery was made, they found that progranulin, its main function is related to the immune system and inflammation. If you breed mice so that they lack the progranulin gene, interestingly, they develop an inflammatory arthritis. If you lack more than half of the progranulin protein that you should have, which is what happens if one of your genes is mutated, you'll develop frontotemporal dementia. If you have a lesser reduction of about 30%, you're at high risk of developing Alzheimer's disease. So I think it would be clear then with that kind of convergence of genetic discoveries, all highlighting the immune pathway, the hypothesis around the relationship between neuroinflammation and microglia and dementia was revisited and is currently still being revisited. So additional epidemiologic studies were done using big data. That's an advantage of the current era. And this time in an epidemiologic study of 41,000 patients with rheumatoid arthritis, they found that 9,000 had Alzheimer's disease. So that was actually a 3% higher chance of having Alzheimer's if you had rheumatoid arthritis than if you did not. However, when they looked at subgroup analysis, they found that patients taking a newer rheumatoid arthritis medication, anti-TNF alpha, actually showed reduced risk of Alzheimer's disease. So again, maybe it's not the systemic inflammation that was key there, but how you modulate it or modify it may influence the risk. Certainly in the clinic where I and Dr. Wells and Dr. Jog see patients with neurodegenerative diseases, time and time again we hear families bring loved ones who were fine and then they had a pneumonia or they had a surgery. They had delirium or a lot of confusion after that serious illness, and they never quite get back to where they were. And many of them actually go on to progress and have worsening of cognitive function until the point where they meet criteria for dementia or neurodegenerative disorder. So there's lots of interest across disciplines, neurology, anesthesia, surgery, medicine, about are there links between inflammation in the body, inflammation in the brain, and dementia. We know that people who've had operations, especially if they're over age 65, very commonly have postoperative cognitive deficits for the first 3 months. There are mixed studies of people who undergo cardiac surgery and go on the cardiac bypass pump as to whether they have an increased risk of dementia after 5 years or not. There's been increasing focus on whether general anesthesia is a risk factor for developing dementia, and I would say if I just thought of my clinical experience anecdotally, I would think that may be the case. But when people really study that formally, it's not yet panning out. It's not showing a specific association at the clinical patient level with anesthesia or a type of anesthetic. What probably is happening there is that there are so many confounds that can't be controlled for that can also be risk factors. So if you have anesthesia, that means you had surgery. If you had surgery, you had cutting of the skin or the tissue in a way that's not normal. So your body's immune system revs up to heal that wound. So is it the inflammation? Is it the anesthesia? Is it differences in temperature during procedures? All kinds of variables that haven't yet been addressed in these studies. In addition, though, to our clinical anecdotal experience, there is some evidence that in animal models of Alzheimer's disease, different anesthetics, and isoflurane in particular, does seem to accelerate the deposition of amyloid and tau like we would expect in Alzheimer's disease. I'll say just one other I didn't have listed there. Interestingly, there's a twin study, so identical twins who share the same genetic background. And there's a study showing that in twins where one person had a serious inflammatory illness, could be infection or autoimmune, compared to their twin that did not, the twin that had the serious infection or inflammation has an increased risk of developing Alzheimer's disease and an earlier age of onset. So a lot of clues in the epidemiologic studies, but the exact etiologies are not sorted out. So this, as I mentioned, is a newer research area for us, but I'm excited to tell you what we're planning. So taking advantage of Canada's first, and I believe still only, hybrid PET MRI scanner at Lawson Research Institute, and the amazing cyclotron facilities that we have in Lawson that can produce very advanced radioligands. Those are the contrast agents that we use in a PET scan that can bind to very specific molecules and light them up in a living person. So these are things we only used to be able to see after someone had died. If we looked at brain tissue, now we can visualize in a safe way while they're living. So this is a picture of the PET scanner at St. Joe's, and this molecule is called FEPA, F-E-P-P-A, and Dr. Justin Hicks in the back is our radiochemist who is a genius in the kitchen of radioligands, has successfully synthesized FEPA for us. FEPA binds to the activated microglial cells in the brain. So microglia can be resting, and we think they're not doing much then. But if they're revved up and actively doing something in the brain, they express a different receptor. So FEPA binds that receptor and can show us in vivo then this one aspect of microglial function. So with that, in our group, we're looking at patients with frontotemporal dementia. For the reasons you saw, as there are many clues to the immune system at least having some role in this condition. So far, we've scanned 4 patients and about many more controls. This is data from Dr. Udana Anizotto, who started this project as a postdoc at Lawson, working with Dr. Keith St. Lawrence, one of the imaging scientists there. She's now hired as a junior faculty, so we're very glad to keep her here in London. But across the 4 patients so far, this is one example. What we see here, the PET, so if you're seeing more binding of the FEPA, the colors are going to be warmer, so red and yellow would indicate the most binding of FEPA, so the most activated microglia. So in our patient with FTD, we see especially in the frontal lobes, the basal ganglia, a lot of binding relative to an age-matched control, which is below, where you see less red and certainly less yellow, particularly in the frontal lobes, which we know are a hot spot of the disease. So we're continuing to recruit for this study. We anticipate that should be wrapped up in the end of this summer or fall, and very eager to see at the group level whether we can see in vivo in living humans abnormal activation of the immune system, which will then be a nice biomarkers Dr. Wells touched on that will allow us to have some index as to as we try different immunomodulatory treatments or different therapeutics, whether we're making a difference in this signal. So I did jump to clinical trials. I forgot my next slide. If only it were so simple. So, maybe the microglia activation is going to be elevated. The FEPA binding is going to be high. It's going to take us a while before we exactly know what to do next. And that's because although I focused on the function of microglia in relation to the immune system and inflammation in the brain, they have a lot of other important roles. So in development, they're very important for development of different aspects of the brain and neural cells. In young adults and adults, they have critical functions for learning and pruning of the synapses, so the connections our neurons make together, that's how we learn. Normally in development, when you've got the right connections, the ones that aren't so useful get pruned away. The microglia are a part of that. And in adulthood, they, of course, have important immune functions. So if you have an infection or a meningitis, the microglia are important for clearing that. So it's not something that we can shut down lightly, and it will even, I think, remain to be seen whether it should be shut down versus revved up, depending on specific patterns and timing of their function. So lots of questions, as I promised, and I hope you'll stay tuned. And in the next year or so, we'll have some more answers. So in summary, probably over time, as genetics suggests neuroinflammation across many of these neurodegenerative diseases, and we're focused on microglia here in London using PET. And I think that's it. Many thanks to all of our collaborators, our research team, many who are here as well.
Thank you so much, Liz, for that incredibly fascinating talk. You truly are on the cutting edge of research, and it's wonderful to hear that Lawson has the technology as well as the people to support you and your research questions. So our third and final speaker of tonight's event is Dr. Mandar Jog. Dr. Jog is a Lawson researcher, as well as the director of the National Parkinson Foundation Center of Excellence at London Health Sciences Centre, the director of the Movement Disorders Centre in London, as well as a professor of neurology at Western University. His research interests include topics such as motor control, neurophysiology, computational modeling, multi-channel recording, and web-based teaching of movement disorders. Dr. Jog has a passion for clinical and scientific innovation and he holds numerous patents that are reaching commercialization with strong collaboration with our university's technology transfer and other industry partners. Please join me in welcoming Dr. Jog to the podium. Thank you.
Thank you very much. So I have to follow two excellent scientific talks to take you out of necessarily the basic science of dementia and change gears to, as you see there, I have an interest in electrical and computer engineering from my background. And so what I'm going to show you today is how we have actually taken questions from the bedside to the bench, solved them at the bench in terms of using technology and some of the things that we need in terms of motor control and motor problems in Parkinson's disease, and taking them back to the bedside. So largely, one of the things that movement disorder people are born with, because we do movement disorders, are video cameras. So we videotape everything. So there are lots of videos which are more likely to show what you want to see in terms of the change in the appearance of patients, and that's what I'm going to do for you today. So one of the unmet needs that we have is that specialists like Dr. Wells or Dr. Finger are very difficult to find. In the movement disorder world, it is very, very difficult for us to have patients come to movement disorder centers like ours. We're the only ones that serve about 3.5 million people in southern Ontario. Toronto is the next center. Similar to this, around Canada, access to having experts that do motor evaluations, very limited. So in my lab, what we've designed, and now reaching hopefully in the next couple of years commercial stage, is one of the first technologies that I'm showing you here, which is a multi-sensor, completely wireless mobile technology, which is basically a bodysuit. And this is from Disney animation kind of thing. And of course, we've written all the software to not animate Kung Fu Panda, but real people. And this is actually a real patient down there. So this is a patient that has put on the sensors and has taken the sensor technology home. And you can see this is a patient with Parkinson's disease. There's reduction in arm swing on one side. You'll see when the patient sits down that there's tremor in the legs. As they're turning, there's turn hesitation. And this is entirely animated from real data. This is not simply animation, as I said, you can see the tremor patient, right? And so using these sensors now, and we have a CIHR grant that we've written recently to be able to make this into a commercial product, hopefully within the year and a half. It already is being used in our lab extensively to be able to generate objective data. You can imagine now, if you're in Collingwood or Owen Sound or Thunder Bay, Sudbury, you can go to the local lab, have the bodysuit put on. It's basically just a Lycra suit, makes you really cool, and walk around in your house, do tasks. All of this is recorded, uploaded to the cloud, and we can analyze it sitting at a desk in London. Not only that, it gives you objective measures for the doctor so that it rates all of the things that we normally do in the clinic, which is tremor, which is dyskinesia, patients with Parkinson's that look like this with Michael J. Fox, gait. All of that is actually provided as a readout to the doctor, and we're almost there. We should be finished that software by the end of August. So that allows the doctor to say remotely, oh, your bradykinesia score is this, your tremor is that, and this and that. Today, this patient would have to travel all the way to see us in clinic after 3.5, 4, 5, 6 hours flying from Thunder Bay, 6 hours of flying time, and in 20 minutes, I'm supposed to assess how they look. And they'd say, I think you're doing quite well, thank you. And you can fly all the way back. Instead of that, this technology is available to do all kinds of things. So we're using this technology now to hopefully, we've already done this work, to program brain stimulation devices automatically to the patient's state, to dose pumps that are being put into patients so that we can have automatic machine-to-machine talk. So the dose of levodopa that is pumped into the stomach is automatically given, and a variety of other things. So this technology is basically smart telemedicine, right? And it's coming to your labs near you very soon. The hope is that this suit, if you were to buy it for Walt Disney, would be $100,000. We buy it for $15,000, not the suit, the sensors. And now the hope is that very quickly, and we've already got the engineering know-how to do this, that'll be $1,000. So you can have a suit. So in fact, for our lab, we ended up having to buy a washing machine because one of the things that we do is take the sensors out. They just stick on like this, and you have to wash the suit. It's that portable. So what it does for us, if this slide advances, hopefully, there, is that you can see in the same patient, as the patient went from an off state with tremor, freezing of gait, and walking difficulty, to developing dyskinesia. So this is all remotely done. We are not actually examining this patient. The sensors are on. The patient took their medication. Before the medication, they were off, sitting with tremor, difficulty with walking, and turn freezing, all animated from real data. This is not just a video animation. This is a real patient. After the drug kicked in, an hour later, the patient became dyskinetic. I don't need to see the patient now. I can say that dyskinesia score is 3 out of 4, really bad dyskinesia. Tremors 2, without examining the patient. This patient could be anywhere on the planet. Right? And so this smart telemedicine, within the next couple of years, will be available. This is developed entirely here at Lawson. Nobody in the world has this. We've done this here. So that's for remote monitoring and to be able to assess and access objectively the measurement of movement disorders in these patients. Any kind of disorders, to be Huntington's patients. Another unmet need is treatment of tremor. Essential tremor affects 4% of the population in the world. That high, right? It's one of the highest movement disorders on the planet. Parkinson's disease tremor is also very similar, but much less in number. Neither of these tremors had any treatment. Parkinson's disease medicines don't work for tremor very well. Neither do the oral medications for essential tremor, which is, as I said, a very common disorder. Botulinum toxin, or Botox that you might have heard of, is a medication that we use quite extensively. I've been injecting since 1990 botulinum toxin for a variety of different movement disorders, including cervical dystonia, post-stroke spasticity, all these kinds of things. The way botulinum toxin works is that it weakens, if you do it right, selectively the muscles that you inject and dampens the activity, hyperactivity of those muscles. Well, then why not inject for tremor? It's easy. Do you have hyperactivity of muscles that flex and extend the wrist, so you can inject those and all good. Problem is, our arms are one of the most complicated machines that exist. You've got 3 joints that are moving in multiple planes simultaneously: flexion, extension, rotation, elbow extension, shoulder. Well, visually, looking at that arm, there's no way, after having done 20,000 injections, I would be able to figure out which muscle groups to inject, in fact, which joints are moving. So, as part of one of the motor control research things that we were doing in our lab, we were using sensors to record biomechanics, how people throw. And this is used in other industries for golf swings and Roger Federer, how to make a serve better and all these kind of things. So we were using this for understanding biomechanics of movement. And lo and behold, we said, wait a minute, we can record the angular deviation at each joint independently, because we designed these special sensors to do this, so that I could record all of the movements of the wrist while simultaneously recording elbow, while simultaneously recording shoulder. And then, of course, being software writing types and mathematics types, we wrote all the software to be able to separate those tremor features out. Not only did we do that, we then went on to decide, based on a learning algorithm, machine learning algorithm, how those joints would be moved by the muscles. Then we injected those muscles, and I have produced the largest experience of botulinum toxin injection using kinematic guidance, or this is machine guidance, for injection of tremor. And this is now being used by Merck Pharmaceutical to design a phase 3 study, first time in the world using a technology, machine-guided treatment of any movement disorder. And in fact, I'll show you, this is on television now. This is a 36-year-old patient with Parkinson's disease. You can see that the upper video is his tremor. The bottom video is moving, by the way. I mean, it's actually working, but he doesn't have any tremor. As you can see, it's not a static picture. That would be kind of cheating. And so, and this gentleman now has been treated for 5 years. He continues to get excellent relief from his Parkinson's tremor, for which you can see how disabling that original tremor would be. We've now treated over 100 and some odd patients, and we have, as I said, the largest experience of tremor treatment using botulinum toxin on the planet. Many, many papers that have been published, and this technology has been commercialized now. It's called TremorTech, and it is a spin-off company which has this product, and Merck Pharmaceutical is going to be using that technology for the final multi-center, multi-continent trial. Again, for the first time, a machine-guided technology is going to be used in the next year with botulinum toxin pairing for treatment of this untreatable condition, tremor. So, remote sensing, using those same sensors. These are different sensors. These are actually, in fact, on the picture here, you'll see that we've gone on from that wired system, which I got to change that picture. That's 5 years old, to completely wireless. So now the system doesn't need any wires. You can walk around and stand. And we've used the same technology for movement of the neck. There's another condition like this as part of our teaching program, which was very humbling, because I trained the system, the sensor system, using my experience to learn how to inject for neck tremor, this kind of a movement. And I think I'm pretty good at injecting. And when we pitched the system, after the machine had learned how to measure the neck movement and how to dose the toxin, it beat me. So it got better results than I got, and so we ended up publishing that. And there was one of the reviewers' commenters said that, what does Dr. Jock think about his clinical ability now? Didn't respond to that review, obviously. Moving from there to treatment of gait. What has this got to do with dementia? Actually, nothing really. But so we changed to try and treat, as I said, the significant disability that is caused by gait. This is freezing of gait in our patients with Parkinson's disease. This patient is walking in the engineering building before we move to our new labs on a gait carpet that has a very significant number of sensors embedded, and you can see that he's not stopping to look. He's actually freezing, and you can see the footfalls down below on this prokinetics carpet, and the footfalls are terrible. I mean, who walks like this? Parkinson patients do. Patients that have severe disability of mobility. In fact, we saw a patient today with Parkinson's disease, not late stages, cognitively completely normal, 54-year-old gentleman, 8 years of Parkinson's disease, who's completely immobile. So we have lots of these patients who are completely immobile. I'm not going to go into the basic neuroscience of what has been done, what we did in our lab in rodent models, to be able to show that if brain control of gait, which is what normally we do, fails, it is possible for us to start talking to the spinal cord, because the spinal cord does have what we call pattern generators. Lots of people have said, well, I mean, I'm not sure it's going to do anything. In fact, there was a very small study that was done in Toronto, which didn't work, and of course, if it doesn't work in Toronto, it must not work. But which they are clearly wrong, because we've shown that it is true. That was Toronto. I trained in Toronto, by the way. They're a very good group and friends of mine. But so what did we do? Well, we used the parameters of this carpet to improve certain things, such as step length, stride width, stride velocity, et cetera, to be able to see if we can actually take technology, data, and program this spinal cord stimulator. Well, what is a spinal cord stimulator? Well, the spinal cord stimulator is this thing. It's actually made by a company called Boston Scientific, and it's used for pain treatment, and it's standard therapy for intractable pain. Or you can use the same electrodes and put them on the posterior part of the spine, on the spinal cord, not in the spinal cord. And it's put over there on the thoracic spinal cord. It takes about an hour to do. It's an outpatient procedure. Patients go home with this thing. We bring them back about a month later, and one of my graduate students, Olivia Samotis, who's doing her PhD on this as part of her PhD, has learned very well how to program. In fact, I actually don't know how to program this thing personally myself. And so she has learned how to do this and has become an expert at this. And we did our first 5 patients, which was published in Movement Disorder Journal, with outstanding results, and I'll show you that. I'm not going to show you the video of the patients, but show you the gait mat, which is everything that you need to see. So here's the patient while on Parkinson's medications, pre-stimulation. So this is how he was walking. I don't think you need me to tell you that this was statistically significant improvement in his walking, right? So this is a normal walk, completely normal. And this is the patient on spinal cord stimulation on levodopa. And in fact, the interesting thing was after we stimulated for 6 months, we have 1 month and 3 months and 5 months and 6 months and year data. I'm just showing you one. This is now at a year that we've got improvement. Even after switching off the spinal cord stimulator, which is the middle graph here, I think there's a pointer here somewhere, but yeah. Oh, wow. Yeah, there we go. Technology. There you go. So if you switched off the stimulator, for 48 hours, his gait continued to be better, right? Unfortunately, it declined after that, but you had to put it on. This second patient was wheelchair-bound for 3 years. Wheelchair-bound, okay? So, yeah. So for 3 years, and in fact, it was funny because I'd seen him for many years, 4 or 5 years before that, and he had a chain that he had put to his shoe. And he would lift the foot and walk like this, and to be able to unfreeze himself. That's the only way he could ambulate even to the toilet. Of course, I thought that, that was a foot drop, because that's quite common in patients who have had other injuries, and, the foot was weak. I never actually examined him to say that he actually didn't have a foot drop. And one of my fellows, Dr. Phil Rizak, who actually did do the right thing and examined the patient, said he actually doesn't have foot drop. I said, what do you mean? Why does he use this chain? He said, yeah, well, that's because to unfreeze himself. We just started the study, so we asked him, you're scooter-bound for 3 years. What do you think? Well, Not only does he not use the scooter, he doesn't use the cane either. He swings his cane if I showed you a video like this. And, of course, for long distances, but this is his gait. At 1 year, he's still walking. 5 patients like this. We've done 10 more after that, and they all have done well, right? And here's the published, this is from Movement Disorders. The balance confidence scale is on top. So these were people who had a confidence scale of walking 2 or 3 steps of less than 40%, are now at 60 and 80%. Not only are they ambulating, they've had no fall. So if you actually look at the improvement in their stride velocity, which is the red bar, all of them are significant, almost reaching significance. But the biggest improvement in their stride velocity, in fact, the first patient's spouse complained, he's walking too fast now, I got to slow him down. This is a person that was not walking homebound, right? So that's not a bad end of result. And here are the freezing episodes. The episodes of freezing went down to 0. Right? So from 16 episodes of freezing per day to 0, right? And so I presented this last 3 weeks ago in Belgium at the freezing of gait meeting, and my talk was at the end. I said, well, all the other 72 talks don't matter really because we fixed it. I didn't say that. I would have been shot. Your work doesn't really matter because we changed it all. Finally, in the end, to show that we actually do science at the level of trying to do discovery, because I think it's important for us not only to bring bedside to the bench, and in this case I've just shown you technology and how technology has been used, but also to address what I think Jenny showed very nicely in terms of the pipeline and how difficult it is to actually get new molecules and drugs to market. So what she didn't tell you is that it costs $1 billion to bring a molecule from the bench, if at all possible. And these pharmaceutical companies go bankrupt, some of them that actually have 1 or 2 molecules trying to develop this. So one of the ideas in the lab that we have, just last 2-3 slides, is how can we repurpose molecules that are already out there, well validated, their pharmacology is known, and apply it to something that actually would shorten that course. And that's taking a molecule like botulinum toxin again, come back to Botox in a way, but this is not Botox, this is animal-grade toxin, and to be able to use the pharmacology of the fact that botulinum toxin not only works in muscles, and I'm not going to go into how it does that, we talked very briefly, but it could be potentially used, using its pharmacological mechanism, to change the chemistry of the brain itself. So botulinum toxin is an inhibitor of release of neurotransmitters. The axons that she showed very nicely release neurotransmitters, which have an effect on neurons and do the things that they do. One of those neurotransmitters that are in the connected network of brain systems in Parkinson's disease is a neurotransmitter called glutamate. That glutamate is released in excessive quantities by excessive firing of a particular group of neurons in a small little nucleus called the subthalamic nucleus, and in fact, that's where we put electrodes when we do brain surgery on patients with Parkinson's disease to, very crudely, let's say, inhibit the activity of those neurons so that Parkinson's patients improve. Well, you have to do surgery and put these big electrodes and do things in patients. That's not really true neuromodulation. So we thought, if we use a molecule, which is the size, I mean, obviously molecule is very small, smaller than the size of the neuron itself, and modulated the activity of glutamate within those neurons, could we reverse the signs of Parkinson's disease? That's exactly what we did. So I'm going to show you 3 animal videos now. We might have to press play there if it's not working, because maybe that one is not automatically playing. There we go. And play. It'll kick in a second. That's a big video. And this is actually a normal animal. It's walking at a normal pace. And you can see that gait-right carpet that I showed you in human. It's called catwalk for some weird reason, but it's actually a rat walk. It was too small for cats. But I think it's with that fashion industry cat. Anyway, so that you can see the 4 paws down there, and that animal is walking normally with equal representation, basically, of the 4 paws. Well, here's the next video of the animal that we made Parkinsonian by giving it something called 6-hydroxydopamine. And this animal has one side lesion, because if we lesion both sides, the animals don't survive. And if that video plays, click. There we go. Yeah. So now you will see that the animal is moving very slowly. This is a Parkinsonian animal, right? So you can see that one side has very long steps, and the animal is taking all kinds of time to walk, right? So this is now a classic model of unilateral lesioned 6-hydroxydopamine lesioned animal. Right? We gave this animal in a particular region called the globus pallidus interna botulinum toxin. 1 dose. Right? And this is 1 month after the injection. Actually, 1 week there's effect. I didn't want to show you all the videos. You can play the video again, please. And you'll see that in a second the animal is walking completely normally. Complete reversal. Right? And so at 3 months, the effect reduces back to baseline. But for 3 months, this animal, 5 days later, for until about 10 weeks, the gait is entirely normal. This is a single dose of botulinum toxin inside the brain. Right? It's very easy to actually do this in human. People think that it'll be very difficult. There are lots of catheter systems out there. We can put a catheter and give botulinum toxin. It's still at the animal stage. The next stage is to do primate. But I wanted to show you that bench science discovery, repurposing a molecule, can be. Maybe it will not pan out, but at least there is some chance that we'll do this. So I wanted to briefly show you, very different than cognition and dementia in terms of motor control, because Parkinson's disease is how you can use technology to be able to solve those problems that we face currently as clinicians, to access remotely specialists, to have unmet needs such as tremor treatment or gait, which are very disabling symptoms, and use technology to try and optimize and treat those things, such as spinal cord stimulation or botulinum toxin, and also repurpose molecules instead of inventing them, which takes a long time, to be able to come to a point where new therapies can be delivered. I'll stop there. Thank you very much.
I think I'm not the only one feeling that we're really in the midst of a true innovator and someone who's not just making incremental changes, but is really making disruptive innovation happen and shifting people's lives in a very dramatic, impactful, positive way.